Welcome to EQ Bloom. Today's journey takes us inside one of the busiest and most vital parts of your cells, the endoplasmic reticulum, or the ER. Often overshadowed by flashier organelles like the nucleus or mitochondria, the ER is where life's most essential tools like proteins and lipids are crafted, folded, and shipped with remarkable precision. From immune responses to hormone secretion, your ER plays a central role in keeping your body alive and well. Let's uncover how this dynamic factory operates 24/7 and what happens when things go wrong. The endoplasmic reticulum is a sprawling, maze-like membrane system extending out from the nuclear envelope. Think of it as a massive production floor attached to the control room, the nucleus. There are two types, the rough ER studded with ribosomes and the smooth ER, which lacks them. Each has its specialty. The rough ER is the site of protein synthesis and folding, the equivalent of a cellular factory line. The smooth ER, meanwhile, is a lipid specialist and detox unit. These membranous folds maximize surface area within the tight quarters of the cytoplasm, allowing the ER to process thousands of proteins and lipids simultaneously. The ER is also physically connected to the nuclear membrane, allowing near-instant communication between your DNA's instructions and the machines that carry them out. Let's zoom in on the rough ER. Its name comes from many of the ribosomes, tiny machines embedded on its surface, which give it a bumpy texture under the microscope. Ribosomes read genetic instructions from mRNA and begin stitching together amino acids into polypeptides, the building blocks of proteins. But proteins aren't useful until they fold into very specific 3D shapes. That's where the rough ER shines. It not only manufactures proteins, but also helps them fold correctly using chaperone proteins. These folding helpers prevent errors and ensure function. The rough ER also begins quality control. Misfolded proteins are flagged early, either corrected or sent for recycling. It's an assembly line and an inspection site in one. Now on to the smooth ER. Sleek, ribosome-free, but just as crucial. It's the cell's main site for lipid synthesis. That includes phospholipids for cell membranes, cholesterol for structural integrity, and steroid hormones like estrogen or cortisol. In liver cells, the smooth ER also breaks down drugs and harmful toxins. Enzymes embedded in its membrane convert chemicals into less harmful forms so that they can be excreted. Another essential job, calcium storage. The smooth ER holds calcium ions and releases them when a signal arrives, especially in muscle cells and neurons. This ion releases trigger muscle contractions, nerve impulses, and even cell division. Without the smooth ER, your body signaling would grind to a halt. Once proteins and lipids are synthesized, they need to be sent to the right location, a membrane, another organelle, or even secreted from the cell. That's where vesicles come in. The ER packages its products into tiny membrane-bound vesicles like shipping containers that bud off and head toward the Golgi apparatus. There, proteins are sorted, tagged with molecular addresses, and fine-tuned. The Golgi adds sugar groups, folds proteins further, or assembles them into complexes. It acts like a shipping hub, ensuring that each protein is correctly labeled and routed. If the ER is the factory, the Golgi is the logistics and distribution center. This ER-Golgi network is vital for all secretory functions in the body, from digestive enzymes in the gut to antibodies released by immune cells. Despite the ER's precision, problems do arise, and when they do, the consequences can be severe. When protein folding goes awry, misfolded proteins accumulate and trigger a cellular alarm system called the unfolded protein response, or UPR. This response tries to fix the issue by halting protein production, increasing chaperone levels, and enhancing degradation systems. But if ER stress continues, the cell may self-destruct via apoptosis to prevent larger harm. Chronic ER dysfunction is linked to many diseases. In Alzheimer's and Parkinson's, misfolded proteins overwhelm the ER. In cystic fibrosis, a singular defective protein misfolds and is destroyed before it can even reach the surface. In diabetes, ER stress impairs insulin secretion in pancreatic beta cells. High output cells like neurons, liver cells, or plasma cells are especially vulnerable. That's why understanding and managing ER stress is now a key area in disease treatment and biotechnology. So what have we learned? The endoplasmic reticulum is far more than a blob in a cell diagram. It's a master craftsman shaping proteins, synthesizing lipids, storing signals, and detoxifying our system. It's the lifeline that connects your DNA's instructions with the physical molecules that keep you functioning. And just like any factory, it needs balance, resources, and maintenance. When ER function breaks down, entire biological systems can fail, from memory to metabolism. Understanding the ER is the key to understanding life and disease at the molecular level. So next time you hear the phrase protein synthesis, remember, it all starts inside this intricate, essential, and awe-inspiring structure. Subscribe to EQ Bloom for more science that reveals how your body truly works from the inside out.